So I just want to thank FormBio for giving me the opportunity to present here today. Kind of happy to see that Jude is not in the front row anymore because it was starting to remind me of my graduate committee, and I was feeling like I was being judged. So maybe he's somewhere out there. I don't know. But I'm going to be talking to you today about how we're working with FormBio, and we've been designing recombinant AAVs and using some of their in silico modeling design software to kind of get ahead of your typical iteration on designing vectors, testing them in vitro, testing them in vivo, and then going back to the drawing board to redesign. So I think this group knows this picture pretty well. It's all seen of AAV vector. We've seen a lot of this from Phil, nice talks by Phil and Brooke and Jude earlier. This is actually from one of Jude's reviews. Just to reorient everybody, you know, you have ITRs, you have your promoter, therapeutic transgene, poly, and then a bunch of other cis elements that you can play with to put in and out of the vector. As I said, this talk is really a melding of gene therapy expertise with bioinformatics, software engineering, data science expertise coming from FormBio, and the gene therapy side of things coming from SolidBio. So this has allowed us to expand our expertise, our basic knowledge, and our infrastructure. So what are the advantages of machine learning? I am by no means an expert in AI and machine learning. Please, I refer you to the FormBio experts for this. But just, I'll briefly touch on it, and that's, analyzing large data sets, as I think we're all aware. We always see these little squiggly lines and stuff, and it's like a black box. You put something in and something comes out the other side, and it's the something going in and the something coming out that we're really interested in. But the magic happens in that black box that I don't understand, and maybe a lot of us don't quite understand. But this does allow us to identify patterns and optimize various parameters, and then large handling of large data-intensive tasks. So when we think about gene therapy, though, and where we want to be able to optimize, we really want to be able to optimize right out the gate. So we'd love to have designed a vector that we know is going to package at 100% and work perfectly, right? And work exactly as we want. So in order to get there, we would like to predict or get some predictions using multiple different permutations. So we'd like to predict at that stage where we have triple transfection, we have a therapeutic vector, and we're modifying that therapeutic vector before we're doing the transfections even, and putting them into cells and producing the vector. And of course, we all know we've heard a lot of talk about getting heterogeneous populations. You can use processing, and you can potentially use other upstream methods to eliminate empty particles and get a more pure drug product so you don't have fulls. And then you also want to do this well before you get into scaling this process up, where it's going to cost you a lot of money once you get into those 2,000 liters and you're trying to do GMP productions. So what molecular species are packaged into vector population? So it's the what and the vector characterization that Brooke just nicely outlined, as well as Phil earlier. So we have, again, our heterogeneous population of vectors. We're interested in characterizing molecular species. So what are the needs? You want a reproducible library preparation. This is really important, and something that I think a lot of us are finding is problematic in the field to some extent. So we'd love to have that a standard, so to speak. We would want reproducible long-read sequencing capabilities, computation and bioinformatics resources, so nice workflows that are repeatable, as well as data provenance with some sort of central access to that data. Solutions, you can do this externally or internally. We work with CROs. That presents a set of its own challenges, so it's something you have to really decide within your own organization. Long-read sequencing at CROs, we do a lot of it, or establish your own internal sequencing capabilities. Establish your bioinformatics pipelines. We're working very nicely as a collaboration with FormBio right now for that. And then where you want to store your data, basically. Are you going to store it internally, build your own servers and your own data storage abilities, or are you going to have cloud access with something like AWS? This is just an example of a streamlined end-to-end workflow with PacBio sequencing and FormBio's bioinformatics platforms. So basically, you're going from your heterogeneous vector population down to the point where you're analyzing your results, as others have talked about earlier. There's various molecular species. I think Brooke is also using form bio, so you've heard a lot of the different molecular species. I'm going to reiterate some of those. But you have things like left partials, right partials, partials, so you get partials in the middle, vector plus backbone chimerics, various other chimerics that show up that you're quantitating, and then backbone products alone. And we're classifying these as single, depending on what type of vector you're using, if it's single-stranded AAVs versus self-comp AAVs. Often you'll see single-stranded show up with self-comp. Really what we're thinking those are like snapback genomes. So some kind of contamination of snapback genomes that's in your product. This is an example from our company of some work we've done where we've looked at the frequency of molecular species. On the left graph, this is kind of a general overview look where we look at, this is a single-stranded AAV vector. You have about 73%. If you might think at this point, well, I have 73% fulls, but not really. Once you look over at the right graph, that decreases quite a bit. You have a lot of snapback genomes. There's a classification that we're calling other, which is somewhat the inability to classify some of the molecules that are in there. And then they can classify host DNAs and then the various helpers, chimeric molecules, and then there's some proportion of the population that's unmapped. But when you take a closer look at that, let me see if I put a pointer here. No pointer. Oh, no. I guess we can't see it on there. Anyway, when you take a closer look at that, the full length is giving about 50%, and then you can see the product over here that you thought was single-stranded is actually comprised heavily of right partials, quite a bit of left partials, and even normal partials. And then going back to Phil's flip and flops, you can get a nice peek at those 1-to-1-to-1-to-1 ratios of the flip and flop that tells you about the integrity of what you've been packaging into your vector. You can map these onto your reference sequence, and what this starts to tell you is exactly where you might be seeing some of these partials, right? And if you know, or you start to get an idea of where some of these partials or some of these truncations are occurring, that can start to say, you can start thinking, well, okay, I need to go in and make a change in my vector at that position to potentially give me a full length. So you can go back and make modifications based on the information that you're learning. Similar to, I think Phil had mentioned this already, you can get a good picture of the host genomes that are being packaged, and these are being mapped. Form bio software helps us to map these to various chromosomes, and specifically we can get an idea if there's specific genes, so we can know if there's particular hotspots that are being packaged from the host cell genome. Okay, so once you know the what of things, you really want to understand, well, why is that occurring? Why am I getting these partials? Why am I getting left partials, right partials, and all this stuff that's happening? Why do I have such a heterogeneous population? So the challenges here are vector design issues that lead to molecular species subtypes. We're trying to identify genetic elements that lead to truncation. So a lot of this is due to truncation. So where can we identify in the vector that can provide us information to change the vector up front? And identifying genetic elements that lead to these snapback genomes and other structures, as well as the software, the prediction tools that we're using with FormBio can give us insight into CPG islands also. So if we're looking to avoid some of those downstream putative or potential immune responses. And then the solutions in silico modeling, which I'm gonna give you more insight into here in a minute. Truncation propensity map across the vector, and then optimizing yield through modeling and CPG modeling. So what might be causing truncated genomes? There's lots of various structures that could be occurring randomly in a sequence, or it could be preventing proper replication of the genome. You could be inserting stuff like a microRNA or something that Phil mentioned, a couple of secondary structures that you could be putting in there that could forcibly cause truncation, so you would have to modify those in a way that would reduce the truncation, so then you go back to a full vector being packaged into your particle. So altered genetic payloads may lead to decrease in effective vector production, which may lead to modified biological outcomes. And that's an important part of all of this, because there's a dearth of research out there on what the biological outcomes are that are actually associated with a lot of these subtypes that we've been identifying. So in silico workflows, again, there's a huge amount of public and proprietary databases that are going into the form site model, all the squiggly lines with all the nodes and everything, and they go through and you put your construct in there, and then out pops some information on construct truncations. You get histograms for that, CPG island detections, as well as CG content, capsid properties. Brooke mentioned a bit of those earlier in her discussion. And then there's some work, I know some initial work being done on being able to modify the constructs in a way that could change expression. So if we take a closer look, as many of us know, we have both a positive and negative strands that are being packaged in these particles, and it's not the case that they're treated equally or that they're equally going to be having the same truncations on both strands. So you may get different truncations based on which strand is being packaged or which strand is being replicated and put in there. So if you notice here, the reverse strand or the reverse replicated strand, those histograms, the numbers are very low on the y-axis for the truncation propensities versus those on the forward replication. And you'll note that this is a CAG promoter, for instance, and there's a lot of secondary structure, a lot of truncation propensity, rather, in that promoter region. So you can go in, you can identify where those are at, and potentially make modifications. Now this is a promoter, so making modifications in a promoter may cause a lot of other problems downstream, so it may be best just to swap the promoter out for one that's more amenable to what you want to do. In addition to this, you can get secondary and tertiary structure probability predictions. So, and then you can take those, the shaded regions that are on there, you can take those, you can overlay those with the truncation propensities. And that'll give you an idea of where you have very strong truncation propensities and as well as secondary structures. And then potentially you can go back and change those secondary, tertiary structures that may be causing problems in your vector. And this is just a histogram showing GC percent content across a vector with CPG percents, and you can see that when you get to a certain point, those shaded regions mark where there's increased risk of potential immune responses that are predicted, not absolute. So because it's a prediction, you may want to go back and say, okay, well, we can go back potentially and change some of those and make modifications in our vector. Again, you can rapidly do an exhaustive in silico analysis to support lead molecule identification. So you can go in and you can make all kinds of modifications to these things up front and get an idea of which one looks the best based on some of these predictions. And then move forward with your top 5 or 10 that you're interested in. So this is kind of a real world example from Solid. Where we have 5 different vectors, so vectors 1 through 5. The GOI is the same, the 3' UTR that we have in there is the same, poly A is the same. Promoters are different for each one of these. And you can see that the top one, we have a promoter, we have a strong truncation propensity up in the 5' end. We have a strong secondary and tertiary region in that promoter at the 5' end. When we change those promoters, and even all 4 of those promoters, when we swap them out, we can see that a lot of that goes away. And when we take a look at that, and we go dive in with our NGS sequencing or long read sequencing, we can see there's a very interesting trend. So with the SSAAV, again, you look at that and you say, oh, maybe we have full length there. And some of it is, probably much of it is in this case, but when you look at the snapback genomes right next to it, you can see that there's quite a difference in that first black bar versus the other. So that vector 1 is the one that had all the truncation predictions and everything in it. And when you look at the right one, when you start to look at the full length, you can see that there's all 4 of those vectors give somewhere in the neighborhood of a 25% increase in the full length vector, while at the same time you're getting a compensatory decrease in the right partials of greater than 17% in all of those vectors. So it's suggesting that the predictions are matching what we would see real world when we start packaging these things. And then this is some real world data of the same vectors, just showing you the predicted truncations and the predicted percent predicted full lengths it's basically taking the truncations and subtracting it from 100%, and you get the predicted values. Those values aren't matching exactly with the percents that we see by real-world NGS, but the trends from vector to vector seem to align pretty well. And then lastly, I just want to reiterate basically that it really is going to be important for us to understand what the biological outcomes are from all of these partials and all of these subtypes that we're putting in there. And I think Brooke started to allude to some of that with her in vitro data that she was looking at. So that kind of data is going to be really important, and understanding if there really is a there that we need to remove and manipulate and get out of it. So you start with these optimized payloads, you do your sequencing, you go into your preclinical animal models or your in vitro assays, and then you iterate on that until you get the vector you want. And hopefully with the predictions, your iterations are going to be a lot less than if you were making 100 different vectors that you're going to need to test to eventually get to a product that would be used as a therapeutic. So just some final thoughts. These in silico tools and partnerships, they can be instrumental to understanding the quality of your drug product and an accelerant for improving the ultimate design of your vector. So I just want to thank basically a lot of the folks at Solid and the 4Bio team for working with us.